Very good evening, all. I hope I am audible. Am I audible, guys? Hope so. I am audible. Yes. So today we are going to, as I promised you in the group, today what we will do is we will first see the animation video of this replication. I will explain you in detail for the first 5 minutes. Then what we will do is we will quickly go through the next set. Okay. Is it clear? Am I audible? Am I audible? Yes. So thank you, Sai Lokesh. Yes, I'm. If I'm audible, I'll just start with the session. So first, what we have to really know is to understand this one particular topic called as replication. So what I can see is most of them have doubts in your replication. That is the reason what I thought is I will just start with this replication topic first. So let us move into this replication. So first, what you need to understand is you need to understand that replication is nothing but replication is a process by which you're just going to find a duplication. You're just going to do a duplication of your DNA strand. Okay, from a parent DNA, what you're going to do is you're going to just duplicate it. So just if you're going to watch this video, so I'll just start with. So see this video. So what you're going to have basically is a replication is a process of copying a parental DNA molecule to form two daughter DNA molecule. Okay, so you're going to just form a two daughter DNA molecule. How we are going to do this? You need certain enzymes for it. As I yesterday explained you need to have certain enzymes for it. So what are those enzymes? These enzymes are nothing but they are going to be called as your helicase. You're going to have your single-stranded DNA binding protein. Then you're going to have topoisomerase. Then you're going to have primase, DNA polymerase, sliding DNA clamps, RNase, and you're going to have a DNA ligase. Now understand, this DNA helicase is the one that is going to cause strand separation. Can you see here? It is going to separate your strand. Once it separates your strand, you're going to get a two single-stranded DNA. So if you have two single-stranded DNA, there is a chances of them clubbing together again. Since they are going to be DNA is going to be a double-stranded, they can come and attach together. So in order to prevent that attraction, in order to prevent that attachment of the single-stranded DNA which are formed, you are going to have a single-stranded binding protein. So this single-stranded binding protein, what it does is it is going to attach with all the single-stranded DNA, and it is going to prevent them from getting recoiled. Now what happens? You're going to get this topoisomerase. What this topoisomerase does, you know that your DNA is a positive supercoiled structure. So what you need to do is this topoisomerase is going to remove the positive supercoil. So now you can see, right? They are going to cut the positive supercoil. Once your supercoils gets cut, you have this DNA primase. What is this primer does? Nothing but you need a induction. So if ஒரு primer எப்படினா உங்களுக்கு ஒரு DNA replication நடக்கணும்னா அதுக்கு ஒரு starting trouble இருக்கு. அதாவது DNA polymerase வந்து டைரக்டா it cannot start producing the copy or your clone of your DNA. For that, what it does is, can you see in this here? The primers are going to be activated first. So இது என்ன பண்ணுதுன்னா ஒரு சின்ன segment create பண்ணுது. So can you see here in the first two, in the both the strands, there is going to be a small amount of starting material. In the starting material மேல உங்க DNA polymerase attach ஆகி it is going to form the DNA synthesis. Very interesting, isn't it? But ஒரே ஒரு problem இருக்கு. என்னன்னா you have two strands. Leading strand ன்னு ஒன்னு இருக்கு, lagging strand ன்னு ஒன்னு இருக்கு. இப்ப leading strand என்ன பண்ணலாம்னா it moves in the direction of 3' to 5'. But lagging strand moves in the direction of 5' to 3'. Therefore, it is very difficult for your DNA polymerase because your DNA polymerase only moves in the direction of 3' to 5'. அப்ப உங்களோட 5' to 3', that is your lagging strand. அங்க எப்படி synthesis நடக்குதுன்னு பாத்தீங்கன்னா ஒரு fragment synthesis நடக்குது. So what happens is, can you see here? So in this video, so first என்ன ஆகுதுன்னா ஒரு fragment synthesis நடக்குது. அதுக்கப்புறம் again ஒரு primer வருது. ஒரு primer comes and attach at one particular point. அங்கேயிருந்து திரும்பி 3' to 5' நடக்குது. So leading strandல single primer தான் required. ஒரு long stretched leading strand can be created. ஒரு long stretched DNA replication can be done. But what happens in your lagging strand? Lagging strandல சின்ன fragments it's going to form. So can you see here? This is your leading strand. Leading strand continuously it is going to form up to 1000 nucleotides it can form continuously. But when you're going to look into your the second thing, that is going to be a lagging strand, it is going to form a Okazaki fragments. Chinna fragments it is going to create. But as I told you yesterday, it is wanted some catalyst. It wants some catalyst to make sure that it is going to go faster. So on the DNA polymerase faster kondu poradhu yaroda vela na sliding DNA clamps oda vela. So ipa sliding DNA clamps enna panudhu fast ah ungalku vandu strands create pandradhukku help panudhu. But unfortunately, what happens? You're going to have this primers. These primers are RNA primers. Appo or DNA la RNA fragments irukalama? It cannot be there. So what happens is this RNA fragments has to be removed. Anga da ungalku RNase oda activity. So what your RNase is going to do is it is going to remove the primers which is present both in your leading as well as your lagging strand. Clear? Leading as well as lagging strand lende RNase enna panadhu, it is going to remove your primers. Once it is going to remove your primers, what happens is, okay, you are going to get the DNA. Once you are going to remove the primers, you are going to get a DNA. But unfortunate thing is, in the DNA oda end vandu you are going to have something called as telomers. So telomers are nothing but they are the ending tail part of your chromosome. And the telomers vandu pathina, it has to be corrected. That is going to be done by telomerase. Before moving to that telomerase step, what happens? Before moving to that telomerase step, what happens basically? You wanted to understand that. So before that, you have this DNA ligase. What does this DNA ligase do? So in the DNA ligase, enna pannu appdin pathina, it is going to do one more important job. So you have one DNA strand, you have other DNA strand. In the replication, multiple DNAs create pannadhu. In the DNAs ella enna pannu, they have to be attached to each other by phosphodiesterase bond. And the phosphodiesterase bond vechi oru DNA இன்னொரு DNA nucleotide link ஆகுதுல்ல, அந்த linking யார் பண்றாங்கன்னா உங்க DNA ligase பண்றாங்க. You should understand this very carefully. Okay va? Once you understand this, everything will become easy. I hope that I have made a very important thing. I have just documented as a, I have just documented it as a video. I hope you all could understand that video. If you have any doubt, you can just ask me. Otherwise, let's move towards next topic, that is going to be your DNA transcription. Before that, we will see the DNA repair mechanism, and let's move towards the transcription. If you have any doubt till now, you can just message in the chat box. I'll just move. If you are clear with that, next I will move towards the next topic. Are you all clear with that? Shall we move towards the next topic? Can someone put it in the chat box? Is it clear? Is the animation video clear? Able to understand? Okay. So with this wonderful day, so with the beginning of this animation, let's move together and towards the next important topic. So whenever a DNA replication is going to occur, of course, there can be some problem in this replication mechanism. That replication mechanism problem is what you call it as a, DNA repair mechanism. So you can have some repairs. So that repair has to be done. So on the repair mechanism, that is your DNA repair mechanism. Upon the DNA repair mechanism, you have two types. Either ஒரே ஒரு strandல problem இருக்கலாம், that is called as your single strand repair mechanism, or it can be a problem in the both the strands, that you call it as double strand DNA repair mechanism. So once again I'm repeating. So in the DNA repairs la rendu type, ஒன்னு single stranded repair la problem இருக்கலாம், or double strand லயுமே problem இருக்கலாம். Single strand la problem இருந்தா you go for 3 type of mechanism, or maximum you can go for 4 types of mechanism. Number one, mismatch repair mechanism. I think I have told you already this in the last class, but I will quickly revise so that it will be helpful to understand. Okay, so single strand repair mechanism, number one, you have your mismatch repair mechanism. What is this mismatch repair mechanism? Guys, please listen to it. So what you have is, you have this is your parent strand. So 5' to T' on the parent strand. In the parent strand, you're going to have this CH3. You're going to have methylation, CH3. In the CH3 is not copied to the children, or you can say it as a progeny. So DNA, you are going to replicate a new DNA, and the pudu DNA strand, what happens is there is no methylation. So if there is no methylation, what you must do, you cannot continue your DNA replication, isn't it? That new DNA is not useful. So what you need to do is, you are going to break that particular area, then you want to replace it. That is your mismatch DNA repair. So on the mismatch we are going to cut it down. We are going to replace with a new one. How you are going to do that? So you wanted some thing to recognize, right? So first there must be proofreading, and then there must be repair. So two mechanisms are involved here. Number one, you need a proofreading. Number two, you need a repair mechanism. Proofreading is going to be done in the direction of 5' to 3'. Appa 5' to 3' nama first sonnola, adu edilla 5' to 3' with respect to that of the parent DNA. Appa newly formed DNA, it will be going in the direction of 3' to 5', 5' to 3'. It will be going in 5' to 3'. Again, I will tell you carefully. So, see here, this is your parent DNA. Okay, you are going to have the new DNA formed. So, parent DNA is going to be in the 3' to 5' direction. But the newly formed DNA will be in 5' to 3' direction. Now, what you have to understand is, the DNA repair mechanism usually goes in the direction of 5' to 3' with respect to that of your parent. So DNA polymerase goes in the direction of 3' to 5'. DNA polymerase, nama padichom, adu 3' to 5' direction la pochu, DNA polymerase. அதே மாதிரியே உங்களோட repair mechanism will be going in the direction of 5' to 3' அப்படின்னு கொடுத்திருப்பாங்க உங்க bookல. But that 5' to 3' direction is with respect to that of the parent DNA. அப்ப the newly formed DNA la how your repair mechanism goes, or how your proofreading goes, remember your proofreading will be going in the direction of 5' to 3'. Okay, your proofreading will be going in the direction of 5' to 3' or 3' to 5'. See, this is your DNA polymerase. The orange one is going to be your DNA polymerase, and the yellow one is going to be your proofreading. Okay, your yellow one is going to be proofreading. With respect to the parent, it is going to be from 5' to 3'. That means that your DNA polymerase goes in the direction of 5' to 3' with respect to your newly formed DNA, whereas your proofreading goes in the 3' to 5' with respect to that of the newly formed DNA. This is very confusing concept. So last and final, I will repeat once. So you have two things: DNA polymerase proofreading. Proofreading and DNA polymerase opposite direction. With respect to your parent DNA, the proofreading goes from 5' to 3', and your DNA polymerase goes from 3' to 5'. Whereas when you take the progeny, that is going to be the daughter DNA. In the daughter DNA, your DNA polymerase goes from 5' to 3', whereas your repair mechanism goes from 3' to 5'. If repair mechanism, what is that called as? It has to remove. Removing exonuclease. Removing exonuclease. So first what you do is, you are going to have your mute TS. This mute DS is the one that is going to recognize the incorrectly paired nucleotide. So mute TS, what it does, it is going to check the DNA. Is there any problem in your newly formed DNA? So problem irundaduna mute TS enna pannuna, first identify pannum. Identify pannite ada mute L kitta sollum. Mute L kitta. So if mute S mute L one agudhu. So mute S mute L one agudhu. Adukapra you have your mute H. In the mute H da enna panna porana, he is going to have this cutting activity. So yaar cut pandranga, cutting activity yaar pandranga na unga mute H. So mute H போறான், அங்க ப்ராப்ளம் இருக்குன்னு கண்டுபிடிக்கிறான், அவன் உடனே என்ன பண்றான், கட் பண்ணிடுறான். அப்ப கட் ஆனவுடனே அந்த இடத்துல உனக்கு கேப் இருக்கும். அப்ப அந்த கேப்பை யார் ஃபில் பண்ண போறாங்கன்னா 2 பேர் ஃபில் பண்ணலாம். ஒன்னு பாலிமரேஸ் 1 வந்து ஃபில் பண்ணலாம், இல்லனா பாலிமரேஸ் 3 ஃபில் பண்ணலாம். பட் திஸ் இஸ் வித் ரெஸ்பெக்ட் டு தட் ஆஃப் யுவர் ப்ரோகாரியோட்ஸ். பட் யூகாரியோட்ஸ்ல ஸ்பெசிபிக்கா டிஎன்ஏ ரிப்பேருக்கு நம்ம யார சொல்றோம்னா பீட்டா polymerase. Okay, you have your beta polymerase. So beta polymerase is the one in your eukaryotes who is going to come and repair that. Who is going to come and repair that? So epsilon polymerase is the one who does the repair, proofreading. Proofreading is done by the epsilon. Proofreading is done by the epsilon. Repair is done by the beta. This is a question which can be asked as a potential MCQ. Beta polymerase in the eukaryotes does proofreading. Okay, epsilon does the proofreading and beta is the one who is going to do repair mechanism. Okay, clear? I'll be asking you all this question once again in the evening of in the quiz, as a quiz in your WhatsApp group. I want you all to correctly answer. ஓகேவா, அதுக்கப்புறம் நம்ம என்ன பண்றோம்னா எக்ஸோநியூக்ளியேஸ் எலிகேஸ் அவங்க எல்லாம் சேர்ந்து வந்து டிஎன்ஏ பாலிமரேஸ் ஆக்ட் பண்ணி ஆஸ் யூஷுவலா உங்களுக்கு ரிப்பேர் பண்ணிடுறாங்க. இப்ப இந்த ரிப்பேர் மெக்கானிசம்ல ஏதாவது ப்ராப்ளம் இருந்தா, மிஸ்மேட்ச் ரிப்பேர் மெக்கானிசம்ல ஏதாவது ப்ராப்ளம் இருந்தா, that will be leading to the one of the most commonest colon cancer pathology la you will be reading next year. So pathology la most common colon cancer is your HPCC, hereditary polyposis colon cancer. This hereditary polyposis colon cancer is caused due to that problem or there is a mutation in your mismatch repair. Is it clear? Next we are going to go for this base excision repair. What happens in your base excision repair? So base excision repair na enna veru oru particular base problem irukku. For example, adenine should go with thymine. Adenine should go with what? Can someone tell? Adenine goes with thymine, guanine goes with cytosine. இப்ப இந்த மாதிரி ஒரு கான்செப்ட் நம்ம வச்சிருக்கோம் சார்காஃப் ரூல்ல. பட் உல்டாவா என்ன ஆகுதுன்னா அடினைன் pairs with cytosine, guanine pairs with thymine, இந்த மாதிரி ஒரு unusual problem ஏற்படுது. So when there is a mismatch in the pairing, what you are going to do is just remove one particular base and you are going to insert with other particular base. So that is called as apurinic apyrimidinic problem. Apurinic apyrimidinic problem. அப்படினா என்ன மீனிங்? apurinic apyrimidinic னா so ஒரு purine pyrimidine base pairல ஏற்படுற ப்ராப்ளம். அப்போ ஒரு purine pyrimidine base pairல ஏற்பட்ட ப்ராப்ளம்னா வெறும் அந்த பர்టిక్యులர் base pair மட்டும் நம்ம ரிமூவ் பண்ணிட்டு then we are going to use the DNA polymerase to insert the new base pair which is correct. Okay, so this is your base excision repair. I hope you can understand this. Very good, Akshaya. So very good, Akshay. So we are going to move towards the next one. So if a first topic nama paathom, that is your mismatch repair. Second, we saw base excision repair. Third, what we are going to see is nucleotide excision repair. So inge ஒரு nucleotide consists of 10 base pair, isn't it? And the 10 base pair me நம்ம என்ன பண்றோம்? We are going to remove it and we are going to replace by entirely new set of nucleotide ஒரு nucleotide. So ஒரு set of nucleotide மொத்தமா remove பண்ணிட்டு 10 base pair புதுசா ஒரு நியூக்ளியோடைடை இன்சர்ட் பண்றோம். அதுக்கு பேர் தான் நியூக்ளியோடைட் எக்ஸிஷன் ரிப்பேர். So whenever there is going to be a problem in the nucleotide excision repair, that leads to two type of disease. Number one, xeroderma pigmentosa, and number two is Cockayne syndrome. So ரெண்டு important என்ன, xeroderma pigmentosa, Cockayne syndrome. இந்த xeroderma pigmentosa is asked recent question நீட்ல கேட்டிருந்தாங்க. and your Cockayne syndrome இருக்குல்ல, அந்த Cockayne syndrome வந்து 2011ல நம்ம ஸ்டேட்டோட என்ட்ரன்ஸ் எக்ஸாம் தனியா நடந்தது, நம்ம ஸ்டேட்டுக்கு பிஜி என்ட்ரன்ஸ் எக்ஸாம், அங்க கேட்டிருந்த கொஸ்டின். ஓகேவா, இந்த ரெண்டு கொஸ்டின்ல இந்த Cockayne syndrome ரீசெண்டா கேட்கவே இல்லை. சோ இதெல்லாம் உங்க யூனிவர்சிட்டி எம்சிக்யூஸா வரதுக்கு சான்ஸ் இருக்கு. ஓகே, தென் this is your xeroderma pigmentosa, guys. What happens in your xeroderma pigmentosa? There is a problem in the nucleotide excision repair. So whenever there is a problem in the nucleotide excision repair, what happens is it is going to lead to this xeroderma pigmentosa autosomal recessive disease. It is a xeroderma pigmentosa. It is a autosomal recessive disease. Once you understand this, I told you clearly that in the beginning, so you have a single strand problem or double strand problem. When there is a single strand problem, you can go for the first three, whichever I told you. But whenever there is going to be a double strand problem, whenever there is going to be a double strand problem, so then what you're going to do? Then you're going to do two things. One, you can go for the homologous repair, that is recombination, or non-homologous end joining repair. So you have two types. Okay, so double strand iron, you have two type of repair mechanism. Number one, you have your homologous repair mechanism or non-homologous repair mechanism. So first one, let's discuss about the homologous and non-homologous repair mechanism. Homologous repair mechanism na rendu irukilla. Okay, rendu DNA strand இருக்குல்ல, அந்த ரெண்டு DNA ஸ்ட்ராண்ட்லயும் similar defect இருக்கு, similar ஒரு error இருக்கு. So ரெண்டுத்துலயும் similar error இருந்தா what we can do is we can cut it actually. So ரெண்டு பக்கமும் கட் பண்ணிட்டு we can cross link them so that they can continue, because ரெண்டுத்துலயுமே ஒரே மாதிரியான ப்ராப்ளம் இருக்கு. So ஒரே மாதிரியா ப்ராப்ளம் இருந்தா நம்ம என்ன பண்ணலாம்? We can actually cross link them. That is called as your cross linkage. At the holiday junction, the place where you are going to cross link, you call it as a holiday junction. Clear ah idu varaikkum? So you can do the cross linkage holiday junction la you are going to cross link it. Okay va, whenever there is a problem in this, so whenever there is a error in this homologous correction, it leads to certain diseases. Number one, you are going to just remember what is that, because you next year, subsequent years la first years will be reading about individual syndrome separately. For now you remember that you will be having a Bloom syndrome, Fanconi anemia, and ataxia telangiectasia. So ataxia telangiectasia. Yeah, ataxia telangiectasia varudhu. There is a spinocerebellar abnormality. So in the balance mechanism irukadhu and the patient ku. So adanala there is going to be ataxia telangiectasia. I hope you are clear with this homologous. Next you are going to have some non-homologous. So non-homologous la enna aguduna, okay, oru idhu innoru idhu same ah irukadu. அப்ப நீங்க ரெண்டுத்தையும் கட் பண்ணணும். ரெண்டுத்தையும் அகைன் you wanted to start with this DNA polymerase ligase எல்லாமே வச்சு அகைன் அந்த பார்ட்டை மட்டும் நம்ம என்ன பண்றோம்? We are going to join together. So what you are going to do is you are going to cut this area, you are going to join these two areas, you are going to join these two areas. This is called as your end joining repair. So in the end joining repair la ஏதாவது problem இருந்தா that will lead to severe combined immunodeficiency, or you will also have the radio sensitive SCID. Now anyone, can you tell me in the chat box if you have seen the first video of this molecular biology? If you have seen the first video of molecular biology, I told you that one particular deficiency of enzyme will cause this SCID, what it is. So what SCID deficiency will cause SCID? Can someone, what enzyme deficiency causes SCID? Come on. So when there is going to be this is actually a problem in the production of purines. Can someone tell what is that enzyme? Adenine deaminase. Adenine deaminase is an enzyme. If it is going to be a deficient, that leads to no DNA synthesis, that leads to severe combined immunodeficiency. I want you all to remember, now you have two important causes of severe combined immunodeficiency. One is due to the problem in the adenine deaminase enzyme, and second thing is going to be because of the non-homologous repair mechanism failure, SCID will be caused. Is it clear? Two important things I told you regarding SCID till now. Now, coming back to the next one, you have certain diseases that are associated with this cross-linking. I told you already, ataxia telangiectasia, Bloom syndrome, Fanconi anemia. Similarly, for non-homologous error, you are going to have this sickle cell anemia. So, non-homologous error correction mechanism la problem irundha, that leads to sickle cell anemia. Is it clear till now? All people, those who are online, those who are watching, can you just put in the chat box, are you clear till now? Because if you are clear with this, next I can move towards the next topic. Okay, shall I move towards the next topic? See, இது வரைக்கும் நம்ம பார்த்தது வந்து ரெப்ளிகேஷன் ஃபுல்லா பார்த்துட்டோம். ஓகேவா? ஃபர்ஸ்ட் 20 minutes நீங்க நம்ம வேஸ்ட் பண்ணிருக்கோம். எதுக்கு? For proper understanding of replication we have wasted 20 minutes. It's okay. So now you all understood 20 minutes la replication fullaவே தெரியும், including the repair mechanism. Now what we are going to do is we are going to move towards the tRNA. Remember, I told you, right? So in the starting of the day, I told that I am just leaving DNA and RNA chemistry. Then what I did yesterday, before starting the DNA replication, we moved on to talk about the DNAs. Today, what we are going to do is, we are going to talk about the RNA. Okay, so now you have this RNA. What is RNA, pa? RNA is just a single stranded molecule. Okay, it's a single stranded molecule. It is going to be not following your Chargaff's rule. And here what happens? Your uracil is going to replace your thiamine. Your uracil is going to be replaced by thiamine. So this entire RNA, what we do is we divide it into two. One is your coding RNA, other is your non-coding RNA. What is this coding RNA? Coding RNA is going to be the one that are going to participate in your coding mechanisms, gene mechanism, protein synthesis mechanism. So who are they? Number one, rRNA. Number two, mRNA. Number three, tRNA. What is this mR and all? rRNA stands for ribosomal RNA. What does tRNA stands for? Transferase RNA. What is your mRNA stands for? Messenger RNA. Come on again, repeat along with me. So rRNA stands for ribosomal RNA, mRNA stands for messenger RNA, and tRNA stands for transfer RNA. So entire RNAs la abundant இருக்கிறது உங்க rRNA. It constitute about 80%, whereas tRNA constitute 15% and your mRNA constitute only 5%, and all are going to be synthesized in your nucleus and your nucleolus. All these RNAs are synthesized in the nucleus and your nucleolus. Okay, wow. So if one structure here, I will show you in detail. So now are you clear with this? Okay, important நீங்க நான் வச்சுக்க வேண்டியது இதுதான், இந்த மூணு தான். ஏன்னா இந்த மூணு தான் they are going to be participating in the coding mechanism. That is the reason why I am giving importance to this particular 3 here. Next we are going to go for the non-coding. Non-coding we are going to have two important things. One is your siRNA, இன்னொன்னு miRNA. What is this siRNA? Silencing RNA. What is this miRNA? miRNA is nothing but your microRNA. Apart from these two, you have two more important things. That is going to be incRNA, inclusion RNA, INC RNA, or otherwise called as inclusion RNA. They are the long microRNA. ஒண்ணுமே இல்ல, microRNA length இருந்ததுன்னா that is going to be called as inclusion RNA. Is it clear, guys? MicroRNA, which is going to be lengthier, you call it as a inclusion RNA. Okay, very good, Aishwarya. It's adenosine deaminase. I appreciate. So this is going to be your inclusion RNA. And you also have your link RNA, that is going to be long intervening ncRNA, that is going to be nano RNA. This is what you need to understand. So after understanding all these things, next what you are going to understand is skip all those things and go to your tRNA in your textbook. மீதி இருக்கிற எல்லா RNA உங்களுக்கு தேவையில்லை. Next, what you have to know in detail is your tRNA. What is your tRNA? tRNA is otherwise called as transfer RNA. tRNA is otherwise called as transfer RNA. What is your transfer RNA? Transfer RNA is the one that is going to undergo your base pairing. So, on the transcription end, what you are going to get is your rRNA, mRNA, tRNA. இதுல tRNA என்ன பண்ணனும்னா translation நீங்க next பார்க்கறப்ப இதுதான் உங்களுக்கு எந்த protein synthesis பண்ணனும் அப்படினு determine பண்ண போகுது. So எந்த protein synthesis பண்ணனும், who is going to determine? Your tRNA is going to determine. Remember, all tRNAs are going to consist of 5 important arms. You need to understand that all tRNA is going to synthesize, is going to be having 5 important arms. Okay, what are those important arms? Number 1, you are going to have a amino acid acceptor arm. You are going to have a amino acid acceptor arm. What is this amino acid acceptor arm? இங்க தான் உங்களுக்கு amino acid இப்ப produce ஆற amino acid accept பண்ற இடம். Okay, who is going to be there? This amino acid acceptor arm. Next, you are going to have your Anticodon arm. So anticodon arm abdina enna, this is the place where it is going to be going and attaching to the mRNA. mRNA la vandu endha codon okkarano abdinga determine pandrathu your anticodon oda vela. Next, you are going to have your D arm, then you are going to have your TC arm. So what are the 4 arms you have? Number 1, amino acid acceptor arm. Number 2, you have anticodon arm. Number 3, you have D arm. And what is the number 4 arm? Number 4 is going to be your variable arm. Variable arm depends upon whether it belongs to the class 1 or whether it is going to belong to the class 2. Whether it's going to be belonging to the class 1 or whether it is going to belong to the class 2, who is going to determine? Your depending upon that, your variable arm is going to determine. So once you know this, next you wanted to know the other one, that is going to be your ribosomal arm. So ribosomal arm la and two type. One, okay, it is going to depend whether it's going to be a prokaryote or it's going to be eukaryote. So ribosomal RNA, it is going to depend on whether it is going to be a eukaryotic or it is going to be a prokaryotic. Okay, depend on these things you are going to see. So prokaryotic arm la eppadi irukum appdin pathina, it is 70S. Okay, prokaryotic is 70S and eukaryotic is going to be 80S. So it is going to be 70S and it is going to be 80S. Clear? So ribosomes, okay, this is ribosome. Okay, ribosome la 2/3 வந்து பாத்தீங்கன்னா rRNA இருக்கு. Okay, மீதி இருக்கிற 1/3 protein. Okay, so in the rRNA are the markers of your ribosomes. So if ribosome is one organelle which is going to be present inside your cell, I hope you all know that. Okay, so இப்போ rRNA குள்ள வந்து பாத்தீங்கன்னா உங்களுக்கு வந்து prokaryotes இல்ல eukaryotes determine பண்ணலாம். Prokaryotes இருந்தா it's going to be 70S, eukaryotes இருந்தா it is going to be 80S. Up to this, is it clear? Next, what you need to understand is each are going to have two important. One is the larger subunit, இன்னொன்னு வந்து smaller subunit. Similarly, your prokaryotes also have two. One it is going to be larger subunit, other is going to be your smaller subunit. Okay, இப்போ large small இது ரெண்டுல எங்கெங்க வரப்போகுது அப்படின்னு பாத்தீங்கன்னா, your smaller subunit of your prokaryote is going to be 30S and larger subunit is going to be 80S. So 50S. So சில பேர் கேட்கலாம், அப்ப ஆட் பண்ணா எனக்கு 70S வராது, 80S தானே வரும். So this is not addition, guys. So this is actually depends upon the size of it. Okay, you cannot add it. So many people will be asking me, so 50S plus 30S has to be 80S and why we write 70S? It's not like that. Similarly, the smaller molecule is going to be your 40S and larger molecule is going to be your 60S. So இது நீங்க மகப் தான் அடிச்சாகணும். Again 80S வந்து 40 plus 60 வந்து 100 னு சொல்வீங்க. It's not 100, it's 80S. So what you are going to remember here? Come on, guys. What you are going to remember? You are going to divide into prokaryotes and eukaryotes. Prokaryotes are going to be your 70S. Eukaryotes are going to be 80S. So in the prokaryotes, what you have? Larger unit, smaller unit. Larger units are 50S. Smaller units is a 30S. Similarly, you have your eukaryotes. Eukaryotes larger is going to be 60 and smaller is going to be 40. So that is what given in this image. Can you see here? Okay, can you see here? So this 60S, 40S, all are going to be nothing but they are going to be depending upon the size and the content of the ribosomal RNA. So இது மட்டும் போதும். இது உள்ள இருக்கிற இந்த minor parts la உங்களுக்கு தேவையில்லை. So I want you all not to remember that. Okay, next what you are going to remember is after completing all these things, then you have your micro RNA and small interfering RNA. This is what I told you. They are going to cause inhibition of gene expression. So they are very important for the therapeutic drug development. So therapeutic drug development la தான் நீங்க அதோட இம்பார்ட்டன்ஸ் பாப்பீங்க. அது வரைக்கும் உங்களுக்கு தேவையில்லை. What is this S? Okay, many people will be asking me, what does this S stands for? S stands for, it is nothing but it's a unit, guys. So you call it as a Svedberg. So if you call it as a Svedberg, it is nothing but it is a unit. Okay, it's just a unit, it is nothing else. More than that, it is nothing else. Just remember it's a Svedberg is a unit. Okay, so idhila edavadhu doubt irukka till this if you have any doubt. So sedimentation rate. Edukku pa sedimentation rate? Can someone tell me what is the sedimentation rate? So Svedberg unit, yes, exactly. This is going to be a sedimentation rate. So 10 power minus 13 unit of time. But and the unit of time வந்து பாத்தீங்கன்னா how it is going to depend upon அப்படின்னா, so that is going to be depending upon your Svedberg sedimentation rate. It is going to be depending upon your length and it is going to depending upon the size. So sedimentation is going to be depending upon size. அதனால தான் what I usually tell is than remembering it as a இது. So what you want to remember is depends upon the size that is going to change. Okay va? So அதனால தான் I equated this size and length as Akshay told. It is like Svedberg is nothing but sedimentation timing. Okay va? This is a sedimentation timing. Okay, just remember like that. But remember length and size base பண்ணி தான் உங்களுக்கு அந்த sedimentation ஆகுது. So அதனால தான் just remember it like sedimentation length and size. அதுக்கு depend ஆனா sedimentation is going to occur. So once I hope you are clear now. If you are okay, I'll just move towards the next one. That is going to be your ribozymes. So I told you have two important things. Okay va? So you have your RNA or the most important ribosomal DNA or the importance enna, you have your RNA molecule that have an intrinsic catalytic activity. That is your ribozyme. So this is going to have a high catalytic activity and they have low toxicity. So your peptidyl transferase, we learn usually enzyme, right? Peptidyl transferase. In the peptidyl transferase is the one that is going to help in transfer of your peptide bond. So other than peptidyl transfer, that is a type of ribozymes. Okay, you can take a screenshot. What you have to know is the difference between your DNA RNA is this 5 points. If you are thorough with this 5 points, you can move towards the next one, that is your transcription. So I want you all to take a screenshot of this particular slide. So after this particular slide, next we are going to move towards our next important topic, that is going to be your transcription. Okay, shall we move towards transcription? Is it clear? Yes. So now we are going to talk about the transcription. So as I told you already, so what we will do is we will first see a video of transcription. After understanding the video of transcription, next we will move towards the entire thing. So let's start. It's a 7 minutes video. So first we will see the video, then we can understand the transcription easily. So see here, I hope my screen is visible. Is my screen visible? Yes. So this is your transcription. A transcription is nothing but it is a process by which your DNA template is copied to an other RNA strand. So first nama paatha replication la enna aguna DNA is converted into another DNA molecule. Inge nama enna pandrona oru DNA strand vechu what we are going to do is we are going to copy it and we are going to form a RNA strand. Okay, listen to this. So can you see here? So ஒரு DNA லந்து ஒரு RNA polymerase என்ன பண்ணுது? It is going to form another RNA strand. So here what happens? You divide your DNA template into different types. So what are those different? So you have a promoter site. You are going to have a exon. You are going to have an intron. So and you are going to at the end you are going to have a terminator site. So first attach அந்த இடம் பேரு we call it as a promoter site and end ஆற இடம் பேரு we call it as terminator site. So you have two types. Okay, you have prokaryotic RNA polymerase and you have a eukaryotic RNA polymerase, very similar to that of your DNA polymerase. So how your DNA polymerase differs for your prokaryotes and eukaryotes, similarly your RNA polymerase also differs with the prokaryotes and eukaryotes. How it differs I will tell you at the last. So now you have a RNA polymerase. This RNA polymerase, what it does is it is going to attach to particular area in your DNA. The particular area at which it is going to attach, that area is called as promoter site. The site at which your RNA polymerase binds to your DNA and initiate a transcription. That site is called as your promoter site. Okay, that site is called as your promoter site. So can you see here? So there is one particular site at which your your RNA polymerase binds with your DNA, template DNA. That site is called as your promoter site. And it ends at one particular point. The site at which it ends is called as your termination site. So first there is an initiation, then there is a elongation, then finally there is a termination. To start with, if you want to have an initiation, what you want is you wanted to form a closed complex. So you wanted to form a closed complex by your RNA polymerase and your DNA template. Then what it must do is it must transit into a open complex. So first promoter site la it should attach. So on the promoter site at which it attaches is there, right? On the site la attach now what you need is you need a sigma factor. The yellow one what you are seeing is a sigma factor. So promoter is a site where your gene is going to, where the RNA polymerase binds it. So on that bind agumbodu, what you need is a sigma factor. What you need is a sigma factor. Then what happens? After formation of closed complex, it has to transit into a open complex. Open complex ah maarnona enna pannona? So DNA open complex ah maarnona and the sigma factor navandukano. So first sigma factor is required for attachment. Then the sigma factor moves so that it gets converted into a open complex. It's going to get converted into a open complex. I hope you can understand till now. So you need, apart from this, what you need is you need a enhancers and repressors. Enhancers now the one which is going to, remember, see here, enhancers are the one that are going to be external agents that are going to increase the transcription, whereas there are certain factors that are going to decrease the transcription. Now I think you can just relate it with your first one, where in the first video we saw that there are two things: methylation, acetylation. Methylation will cause repression of your DNA transcription, whereas acetylation will cause increase in the transcription, activation of your transcription. Adha enhancers and repressors which I am telling. Now what happens? You have a promoter site. This site is the RNA polymerase is going to come and attach. This RNA polymerase is going to do a transcription. So RNA polymerase, like your DNA polymerase, it goes in which direction? So that we will see now. See, abortive initiation. Now the period during the transcription initiation, in which your RNA polymerase repeatedly synthesize short RNA molecule. So chinna RNA molecules synthesis panno, but at abort side. So it is going to stop. Okay, it's going to form only small one. And after one particular point, it is going to continue to form a bigger RNA particle, bigger RNA strand. Can you see here? So at this particular point, there is more RNA strands that are going to be formed. Then you have this promoter escape. So promoter escape na the particular point at which promoter thandi poidudhu. It is going to move away from your promoter. So if a promoter, what is that? Anyone? You have two types of promoter. Okay, I'll explain you in detail when I am taking. Anyway, we will complete the animation first. For now, I just wanted to tell you that promoter site prokaryotes la vera madri irukum, eukaryotes la vera madri irukum. Prokaryotes la Tata box, eukaryotes la you have this cat box, CAT box. Okay, this alone you remember now. And another one point I want to tell you is now we have completed initiation phase in your video. Initiation phase la again I am repeating, there are 4 points. Number 1, formation of closed complex. Number 2, you have transition to open complex. Then you have an abortive initiation. And finally, promoter escape. These are the 4 steps which we have seen in the initiation phase. Next, let's see. This is the steps of elongation. Elongation, la entire thing is going to be done by your RNA polymerase. RNA polymerase is going to continuously, it is going to synthesize your RNA. Then finally, RNA proofreading will be done. After proofreading, it has to dissociate RNA. The RNA comes out. Okay, it has to dissociate. And once it dissociates, what happens? Same way. Okay, eppadi unga replication irundho. Can you see here? Can you just understand this concept? So, eppadi ungaloda replication phase la, nama vandu we saw DNA helicase paathomo. Adhe maadhiri ungalku DNA unwind pannu. Ena namakku ore oru strand da nama template ah eduthukurom. Next enna agudhu? You have your synthesis of RNA. Then you have RNA proofreading. Then after proofreading, that RNA has to come out. That is dissociation of RNA. Okay, RNA dissociate ஆகுது. Dissociate ஆனோனா உங்க ரிமைனிங் இருக்க DNA டெம்ப்ளேட்ல நம்ம அன்வைண்ட் பண்ணிருக்கிற டெம்ப்ளேட் என்ன பண்ணனும்? திரும்பி கன்சீல் பண்ணனும். That concealing is similar to that we done in your replication. So எப்படி நம்ம ரெப்ளிகேஷன்ல திருப்பி அந்த ரெண்டுத்தையும் வந்து கன்சீல் பண்ணோமோ அதே மாதிரி at the end we have to re-anneal. Who is going to do that re-annealing? DNA ligase is going to do. DNA ligase is going to do. So can you, up to this, is it clear? So you have your RNA proofreading. So இப்போ ஒருவேளை incorrect nucleotide இருந்ததுன்னா அதை என்ன பண்ணும்? அந்த இடத்தை கட் பண்ணிடும். அந்த இடத்தை கட் பண்ணிட்டு அதை அப்படியே கட் பண்ணி வெளில தள்ளிடும். Again it is going to start forming the new nucleotide. Again it is going to start forming the new nucleotide like the one which you are seeing. So this is called as the proofreading. So proofreading பொறுத்த வரைக்கும் you have 2 type of proofreading. One pyrophosphorylatic proofreading, இன்னொன்னு you have this hydrolytic editing. So what are the two type of proofreading? Again, I will repeat all this. At that time you need to answer me. Number one, you have your pyrophosphorylatic editing. Number two is your hydrolytic editing. Hydrolytic editing is done by Gree factor, GRE, Gree factor. Okay, can you see here? This is your RNA polymerase, which is going to start doing your proofreading. இங்க வந்து proofreading க்கு separate எந்த enzyme கிடையாது. So இங்க proofreading வந்து same your RNA polymerase is going to do. Can you see here? Phosphorylatic, pyrophosphorylatic editing. So pyrophosphorylatic editing என்ன ஆகுது? So first வந்து it is going to form. Can you listen? See here carefully. First, there is a wrong nucleotide attachment, and the nucleotide உடனே வெளில தள்ளிட்டு new nucleotide form பண்ணிடும். So that is your pyrophosphorylatic editing. Whereas what is a hydrolytic editing? You will see that. Okay, next it is coming. So what is your hydrolytic editing? So hydrolytic editing வந்து incorrect nucleotide இருந்தா you have a Gree factor. That Gree factor is going to help in cutting down that nucleotide, throwing away, then continue your DNA, RNA polymerase activity for synthesis of RNA. So in your proofreading, simultaneous நடக்கும். That is what I want to tell. So how your DNA polymerase, which done in your DNA replication, proofreading at the end நடக்குது. அந்த மாதிரி இல்லாம elongation ஆகும்போதே proofreading நடக்கும். That is what I want you to tell. And finally, what happens? Termination. Termination, there are two types. Either it is going to be row dependent or row independent terminators. Okay, row dependent terminators, row independent terminators. Independent is nothing but intrinsic. Can you see here? Row independent intrinsic terminator. So you have a stop codon. So this is an inverted repeat. So can you see here? So you have an inverted repeat. What you are going to do is just see there. What you are going to do is you are going to just check, and when there is a stop codon, what you are going to do, when there is a stop codon, it is going to stop. Okay, when there is a stop codon, it is going to bind it. Okay, there is going to be a binding and it is going to stop. So and the binding பண்ணிட்டு நிறுத்துறதா வந்து பாத்தீங்கன்னா row independent. So 20 nucleotide inverted repeats இருக்கும். So இந்த மாதிரி inverted repeats இருக்கிறப்ப கட் ஆயிடும். அது row independent terminators. Then what you have is a row dependent terminators. What is row dependent terminators? Row dependent terminators are nothing but, can you understand what is row dependent terminator? Just see there, you have a row factor. This row factor comes and attaches to the particular place where you have to stop, and the row dependent terminators என்ன பண்ணும், அங்க வந்து attach ஆகி what it is going to do is it is going to stop that particular place. It is going to cut down at that particular place. So this is your row dependent terminators. I hope I am making sense and I hope you all understand. Okay, so this is your entire transcription. So it starts with RNA polymerase, primers, which is first you have your initiation phase, then you have elongation phase, then you are going to have termination phase. In the madri, multiple RNA polymerase can transcribe the same gene at same time. So a cell can synthesize large number of RNA transcript in a short period of time. In a short period of time. With this, actually, we are completing this entire RNA transcription. We are completing this entire RNA transcription. If you have any doubt till that point, you can just ask. Otherwise, let's go with the theory. Okay, so now we have completed the video, and video I think hope you all understood something from this video. Some you can just comment in the chat box. So once you understand that, next what we will do is we will just quickly go through the theory part so that it will give you some more understanding and some more points whichever we have left, we will be able to add it. So now again tell me, what are the different what is transcription basically? So you have the cell dogma. Okay, you have the cell dogma. So where the replication occurs, after replication, you wanted to synthesize a mRNA. For that synthesis of mRNA, you do this transcription. So then after transcription, you will get a mRNA. And the mRNA enna pannuna synthesizes new protein, that is called as translation. That is called as translation. Remember, unlike your replication, transcription occurs all the time. So இப்பயும் நம்ம பாடிக்குள்ள நம்ம செல்ஸ் transcribe பண்ணிட்டு தான் இருக்கு. So அதான் transcription. So to run through quickly what we have understood. So we have this RNA polymerase. Yes. So RNA polymerase what we have is two factors இருக்கு. ஒன்னு alpha beta, இன்னொன்னு we have is a sigma. Alpha beta is called as your core protein, whereas your sigma is a alloenzyme. So in the sigma என்ன பண்ணுதுன்னா, this is going to help in initiation of transcription at the promoter site. This was asked one time as a MCQ in your NEET PG. Okay, Then this is your promoter site. At the promoter site, it is going to bind specifically. And there are many type of promoters, as I told you. You have Tata box, you have a Shine-Dahlgren sequence, you have a Goldberg-Ognes box, or you have different types. So in the Tata box, prokaryotes, we call it as Pribnov box, and in eukaryotes, we call it as Ognes box. So idella upload detail. What you want to know is that promoter site we call with different names. Tata box no na solrom, innone we call commonly as CAAT box. Okay, va? So what is the difference between your Tata box and CAAT box? So Tata box ko CAAT box ko difference enna, Tata box and it is going to be doing it is going to be initiating the tRNA synthesis. It is going to initiate in the transcription by 10 position upstream. By 10 position upstream. Whereas other one வந்து பாத்தீங்கன்னா 15 position upstream. So அந்த மாதிரி எப்படி அந்த first set form ஆகுதோ அதை பேஸ் பண்ணி we call it as Tata box, we call it as cat box. Okay va? So எந்த sequenceல it is going to go and attach. அதை பேஸ் பண்ணி தான் நம்ம சொல்றோம். பட் இவ்ளோ தெரிஞ்சா போதும், இதுக்கு மேல தெரிஞ்சுக்கணும்னு அவசியம் இல்ல. Next we are going to move towards the steps in initiation. So steps in initiation, remember your 4 steps. Number 1, it has to form a closed complex by the help of sigma factor. Number 2, it has to form a open complex. It has to transmit to an open complex. Number 3, abortive initiation. And number 4 is promoter escape. So 4 point clear. Then you have elongation, RNA polymerase activity, proofreading. So proofreading is of 2 types. What are the different types of proofreading? What are the different types of proofreading? You have a hydrolytic proofreading and you have the pyrophosphorylatic proofreading. Hydrolytic proofreading and pyrophosphorylatic proofreading. Then you have termination. You have row dependent termination, row independent termination. Okay, well, I hope so. Up to this, you all are understood. Okay, up to this, if you have any doubt, you can put. Otherwise, let me move towards the next one, that is going to be what is the difference you are going to see in the transcription occurring in the eukaryotes. So difference on the pathagana. So RNA polymerase activity, promoter region initiation, either difference I told you. The last one is going to be modification. So in the modification, eukaryotes this is the difference between a prokaryotic transcription and eukaryotic transcription. In a mammalian RNA polymerase, you have type 1, type 2, type 3. Type 1 is responsible for rRNA synthesis. Type 2 is responsible for mRNA synthesis. Type 3 is responsible for tRNA synthesis. This I want you all to remember. RNA polymerase, 3 type. Type 1, type 2, type 3. Type 1 is helping in synthesis of rRNA. Type 2 helps in synthesis of mRNA. Type 3 is helpful in synthesis of tRNA. So I told you that all these 3 are important for the translation to occur. Appa idu 3 synthesis agum in the mammalian RNA. Okay va? So the promoter side. As I told you, promoter region in the eukaryote, it is going to be Ognes. Tata box on the Ognes, cat box on the it is going to be CAD. So now you can ask me what is Ognes, Tata box, and what is cat box. So in your so it in sequence at the promoter region, it is going to be cause you can see a Tata. TATA, adana the Tata box and solrom. Can you see a CAAT cart box? Adana we call it as cart box. Now you understood, right? So what is Tata box? What is cart box? Everyone are going to be a promoter regions. That's all. Now promoter regions, it is going to be Ognes box or cart box. And you have to know the differentiation point in the initiation. So initiation, at least 7 transcription factors are there, which we collectively called as transcription factor 2. So Tata box is recognized by Tata binding protein. Okay, TBB, Tata binding protein. And next what you need to remember is, you need to remember the post-transcriptional modification. Finally, we are moving towards the very important part, that is going to be post-transcriptional modification. So what is this post-transcriptional modification? This is going to be nothing but mRNA which is formed. You call it as primary transcript. Now you have a mRNA, a precursor, that is called as a primary transcript. Now that is called as your primary transcript. But what happens is they don't have this capping, they don't have this adenylation. So capping, adenylation you need for stabilization of your RNA. So for stabilization of RNA, you need to do some post-transcriptional modification. So that is what we are going to see. And similarly, now I want to tell you what is introns and what is extrons. So if you have RNA sequence, so this is going to be a promoter site. This is going to be called as your promoter site. And at this particular point, you're going to have a start. Okay, start synthesizing இந்த இடத்துல நடக்கும் உங்க DNA. இது எல்லாமே உங்க DNA நான் சொல்லிட்டு இருக்கேன். அதுக்கப்புறம் you are going to have exons, then you have your introns, then you have your exons, then you have your introns, and at the end, after some millions of things, then you are going to have this termination site. This is going to be a termination site. இதுதான் நம்ம என்ன பண்றோம்னா we're going to copy it into your RNA. So அப்ப RNA என்ன இருக்கும்? You have your extrons, you have your introns, you have your exons, you have your introns. So நம்ம என்ன பண்ண போறோம்னா உங்களோட precursor RNA which is going to contain this exons, introns, we are going to remove all the introns away. Exons மட்டும் தான் வைக்க போறோம், introns எல்லாம் remove பண்றோம். Why? Because introns are nothing but they are waste products. Exons are coding sequence which is going to help in production of protein, but your introns, they are waste products. You wanted to remove that waste junk material from your mRNA, which is a precursor mRNA formed out of this transcription. So அந்த மாதிரி remove பண்றோம் பாத்தீங்களா, அந்த removalக்கு பேர் தான் post transcriptional modification. ஒருவேளை இந்த introns இருந்ததுன்னா what happens is there is a more number of mutations that can occur. So இந்த mutations prevent பண்ணுங்கிறதுக்காக நம்ம என்ன பண்றோம், introns remove பண்ணி தூக்கி போட்டுறோம். Now I will tell you one by one what are the post transcriptional modification which you are going to have. Number one, remember, you will be doing number one, what you will be doing is you will be having this splicing. So splicing no பண்ணுவீங்க. What is this splicing? Splicings of exon. So what you will do in the presence of spliceosomes, you will just remove this exon. That is called as your splicing. What is capping? So what you're going to do is at the 5 terminus, you're going to cap it by the 7-methyl guanosine triphosphate. 7-methyl guanosine triphosphate வச்சு இந்த இடத்துல what you're going to do is you're going to put a cap. So this cap is the first step which you are going to do at the 5' end. This is going to be a 5' end. This is going to be a 3' end. At 5' end, what you're going to do is you are going to put a cap. What cap it is? 7-methyl guanosine. 7-methyl guanosine. அதே மாதிரியே எட்டை நம்ம கவர் பண்ண மாதிரி டெய்லையும் நம்ம என்ன பண்றோம்னா we are going to stop it. So what you are going to stop at the tail by using a poly A tail, polyadenylated tail. Polyadenylated tail. What is this polyadenylated? A இது மாதிரி adenyl multiple A இருக்கும். ஓகேவா? A to the power of n ன்னு போட்டுக்கோங்க. Multiple A's இருக்கும். Almost 1000 A இருக்கும். So அது வந்து என்ன பண்ணா tail endல attach பண்றோம். இது ரெண்டு எதுக்குன்னா for providing the stability for your RNA. What I have given two things. ஒன்னு வந்து cap, இன்னொன்னு வந்து tail. Cap என்னது? 7-methyl guanosine. Tail என்னது? Polyadenylated tail. So why you are going to put this? For giving a stability for your RNA which is formed. Up to this, is it clear? Next I am going to move towards what is that is called as your methylation. What you are going to do by naming methylation? So you are going to methylate the N6 of adenine residue and 2-hydroxyl group of ribose. This is cytoplasm occur agum, but this is not very important. This is very important. This is called as your splicing. This is called as your splicing, where you are going to remove all the introns. So you are going to remove all the introns. Once your introns are removed, you are going to call that as a mature mRNA. Once your introns are removed, you call that as a mature RNA. You are going to call it as a mature RNA. Can you see here? This is a mature RNA. You are going to have just your blue color part is your exons and white color part is your introns in this picture. So only exons are going to be there. Introns are going to be removed. Introns are junks. Once you are going to remove the junks, you are going to form a mature RNA, mRNA. But in the mature mRNA, again what you are going to do is you are going to do certain important concepts. What are the certain important things what you are going to do? Okay, number one is going to be mRNA editing. So mRNA editing is done by two important materials. What are the two important materials which are going to do mRNA editing? mRNA editing is done by ApoB48 and ApoB100. Okay, similarly you have this gene duplication mechanism. This gene duplication mechanism of the bacteria are what creating antibiotic resistance. நம்ம காமனா சொல்றோம்ல, so antibiotic resistance அப்படிங்கிற இந்த டாபிக் இப்ப ரொம்ப வைட் ஸ்ப்ரெட் டெவலப்மெண்ட் ஆயிருக்கு. So அதுக்கு காரணம் யாருன்னு பாத்தீங்கன்னா gene duplication. Similarly you have this gene rearrangement. Gene rearrangement, why it is done is in order to control this jumping genes. In order to control this jumping genes, you are going to have this gene rearrangement. Till this, do you have any doubt? If you don't have any doubt till this, next I want to talk about this abortive initiation. So do you all remember the term abortive initiation, a period during your transcription initiation in which RNA polymerase is repeatedly synthesized into short RNAs, and the short RNAs synthesis and the short RNA is nothing but that is called as short RNA is the one that is called as what? That is called as naked RNA. So the product of your abortive initiation, that is called as abortive initiation is called as naked RNA. It is called as naked RNA, otherwise called as nascent RNA, or we can also call it as a preliminary graft. Okay, till this, do you have any doubt? If you have any doubt till this, you just let me know. With this, we are completing the transcription. With this, we have actually completed the transcription. Once again, I repeat all the post-transcriptional modification. So you're going to, first what you're going to do, you're going to add a 5 cap addition. So first what you're going to do, you're going to add 5 cap. Then what you're going to do, you're going to give 3 polyadenyl tail. So after 3 polyadenyl tail, what you're going to do, you're going to do splicing. Then what you're going to do, you're going to do RNA editing or differential RNA processing or chemical modification of RNA. Why do you do all these things? I told you, why do you remove the introns? Why do you add 5 cap? You add 5 cap to provide stability. Why do you add 3 polyadenyl tail? In order to prevent itself from the action of exonuclease. And this adenylated tail will help you to pass from your nucleus into cytoplasm. So this adenylated tail, it has to go into the cytoplasm to continue your translation. Because you want to continue your translation, because of that, you have this polyadenylated tail. Okay, then you know what is splicing, then you know what are the differentiation or what are the editing. Now, after understanding all these things, next you are going to talk about this gene expression. Can we go into the gene expression, guys? Okay. So what is gene expression? Just remember, this is a weekend, so we will have some more time. Okay, so we have some more time. Once again, abortive initiation, sir. Okay, so I'll tell you once again what is abortive initiation. Abortive initiation, I think, I hope so you haven't seen that video. So adula enna nama paathona first, okay, ஒரு டிரான்ஸ்கிரிப்ஷன் நடக்குதுன்னா ஃபர்ஸ்ட் உங்க ப்ரொமோட்டர் சைட்ல RNA பாலிமரேஸ் வந்து பைண்ட் ஆகுது. பைண்ட் ஆனோம்னா உங்க சிக்மா ஃபேக்டர் ரிமூவ் ஆகுது. சிக்மா ஃபேக்டர் ரிமூவ் ஆகணும்னா ஓப்பன் இனிஷியேஷன் வந்து இட் இஸ் ஃப்ரம் யுவர் க்ளோஸ்டு காம்ப்ளெக்ஸ் இட்ஸ் கோயிங் டு கெட் கன்வெர்ட்டட் இன்டு ஓப்பன் காம்ப்ளெக்ஸ். பட் ஓப்பன் காம்ப்ளெக்ஸ் ஆனோம்னா உடனே உனக்கு வந்து பாத்தீங்கன்னா RNA வந்து ஃபார்ம் ஆகாது. ஸோ ஃபர்ஸ்ட் 10 நியூக்ளியோடைடா சின்ன சின்ன RNA ஃபிராக்மெண்ட்ஸ் ஃபார்ம் ஆகும். பட் இட் இஸ் நாட் ஏபிள் டு ஃபார்ம் அ பிக்கர் RNA ஸ்ட்ராண்ட். ஸோ அப்போ உங்களுக்கு சின்ன சின்ன RNA strand ஃபார்ம் ஆகுது இல்ல, that small RNA strand is called as abortive initiation process, and the strand which is formed, that is called as naked RNA. That strand is called as naked RNA. Once it escape your promoter, so அந்த promoter regionல இருக்கிற வரைக்கும் உங்களுக்கு இந்த small fragments form ஆகும். Once it escapes out of your promoter fragment, once it escapes out of your promoter site, it is going to form that complete RNA strand. அப்பதான் elongation phase start ஆகும். Initiation phase ல தான் formation of closed complex, open complex, abortive initiation and promoter escape. So after promoter escape, you are going to move towards a elongation where RNA polymerase is going to act. Then you have your proofreading. Then comes a termination site where it can be either row dependent or row independent. I hope so you understood this, Aishwarya. Can I move towards the next one? That is going to be your gene expression. Okay, how you are going to regulate this gene expression? Very, very important. Regulation of gene expression. That is what we are going to discuss at the next. Okay, so in this weekend, we are going to make it very worthful that after completing your replication, after completing your transcription, this is the point where you need to understand, okay, how this genetic expression is going to be regulated. So genetic expression eppadi regulate pandrom abingadha we wanted to say. I hope Aishwarya is clear with this abortive initiation. Okay, now I'm going to move towards the genetic expression. So this genetic expressions and regulation, it is going to be different in prokaryotes and it is going to be different in the eukaryotes. Just write it down. It is going to be different in the prokaryotes and it is going to be different in the eukaryotes. Okay, eukaryotes. Okay, prokaryotes on the pathangana, it is going to be following this lac operon. So on a book lac operon, we will be all confused. What is this lac operon? What is this going to do? Why I have to read all these things? There is one important reason why you are reading all these things. So if you're going to apply it in the clinical way, it is going to be very easy for you all. So prokaryotes, eukaryotes, this lac operon concept, it is given by Jacob. So it is given by Dr. Jacob and Monod. So they are the one who gave this model of lac operon. And eukaryotic, I hope so. If you have seen my first video, I told that all your DNA, they are going to be packed together to form chromatin. And this chromatin is of two types. Either it can be euchromatin or it can be heterochromatin. This euchromatin, basically it increases your transcription, whereas your heterochromatin, it will decrease your transcription. Heterochromatin will decrease your transcription. Now, let's move into this concept called as operon. Let's move into the concept called as operon. So, okay, so operon போறதுக்கு முன்னாடி RNA synthesis ஓட inhibitors அப்படியே பாத்துருங்க. So, beta subunit of RNA polymerase inhibit பண்றது யாருன்னா rifampicin. So, your rifampicin drug inhibits your beta subunit of RNA polymerase. And your actinomycin D, mitomycin, they're going to bind with your template strand of RNA, of your DNA, and they will be blocking your transcription. Similarly, alpha amantadine, it is going to block your RNA polymerase 2 as well as your 3. Block panno, and your deoxyadenosine chain termination, it is going to by incorrect entry, it is going to block. Okay, now let me talk about this lac operon concept. So lac operon, it's nothing but I'll just draw a gene along with me, just try to draw that. Okay, so why you are going to read all these things? All these things are only called as epigenetic. What is epigenetic? Anyone? Epigenetic is nothing but when you're going to deal the in-depth concept of your genetic strands, okay, and the strands of genetics நம்ம வந்து படிக்கிறோம்ல, அதுதான் epigenetics ன்னு நம்ம சொல்லுவோம். So அந்த epigenetics அதான் நம்ம indeed we are going to see today. So what happens is, okay, you have a lac operon concept. What is this operon or what is this cistron? So they are nothing but functional unit, okay, or a basic functional unit of a gene is going to be termed as your cistron. Okay, is termed as your cistron. So you have a mRNA, right? Okay, you have, what is the end product of this transcription? So end product of the transcription is going to be mRNA. You're going to have tRNA, you're going to have rRNA. Among them, I will tell about this mRNA. Sir, what is the trick to remember inhibitors? See, inhibitors வந்து பாத்தீங்கன்னா எந்த இடத்துல என்ன அப்படின்னு நான் வச்சுக்கோங்க. So similar, for example, RNA polymerase the first step இல்ல, அங்க inhibit பண்றது உங்களோட rifampicin. At the end of the day, what you have is RNA polymerase 2, அதை inhibit பண்றது alpha amantadine. So RNA தவிர what you need? DNA template. அந்த DNA template inhibit பண்றது யாரு? Actinomycin D. So you're going to remember like this only. There is no specific way to remember. So mnemonics, certain areas it will work. But in the area, understanding the concept and writing is more easier than understanding the mnemonics and other things. Okay, so I'll tell you wherever mnemonics is required, I have written and kept, I'll tell you. Okay, so now coming back to the basic functional unit of gene, that is your cistron. So the end product of your transcription is going to be your mRNA. End product of your transcription is going to be your mRNA. Okay, in the mRNA, it is of two types. One, polycistronic mRNA. Okay, you're going to have a monocistronic mRNA. Before that, can anyone who is in this session, can you tell me what is this operating unit or your operon model suggest? Operon model enna suggest பண்ணுது? Can someone tell me if you have listened to your classes in your regular lectures? or anything. If you have any idea about this operon concept, you can put it in the chat box. I'll tell you. Monocistronic mRNA in the mRNA வந்து monocistronic எங்க இருக்கும்னா monocistronic mRNA, it will polycistronic mRNA is present in the prokaryotes, and monocistronic mRNA it is present in the eukaryotes. Remember this cistron nale what is the meaning, or your operon na what is the meaning is it is nothing but switch. What it is? It is a switch. So எப்படி fan on off பண்றதுக்கு switch வச்சிருக்கோமோ அதே மாதிரி இந்த cistrons are nothing but they are going to be a switch. Okay, they are going to be a switch. They are going to be seeing whether the this reaction has to be done or reaction தேவையா தேவையில்லையா அப்படின்னு பார்க்குற ஒரு switch தான் this operon. So okay, now what you do is, now remember you have a gene. Okay, so in the gene பார்த்தீங்கன்னா expresses 24/7. It is going to be expressing for 24/7. Appa 24/7 express aite irukna, you need some housekeeping genes. What you need? You need some housekeeping gene in order to make sure that right proteins are synthesized in your body, right enzymes are synthesized in your body. So adudha unga expression வந்து control பண்றதுக்கு help பண்ணுது. So you have something called as housekeeping genes. Similarly, there is something which is going to express only when necessary. So there are some genes which is going to express only if necessary. Okay, only if necessary. So there are two types of gene. One is going to express 24/7 into 360 days a year, and there is some gene which is going to express only if it is necessary. Okay, the one which is going to be doing only if it is necessary or otherwise it is going to be silent, that is called as your inducible gene. That is called as your inducible gene. Okay, for example, you have your yeast, isn't it? You have your yeast. So yeast la மூணு enzymes இருக்கு. என்னென்ன? beta galactosidase ஒரு enzyme இருக்கு. Second thing, you have your permease and you have your acetylase. Okay, இந்த மூணு ஜீன் வந்து பாத்தீங்கன்னா they will be expressing only when necessary. Okay, only when necessary. So that is called as your expressive gene or inducible gene. For example, okay, so இப்ப இந்த yeast வந்து பாத்தீங்கன்னா எப்ப it is going to act அப்படின்னு நம்ம பார்த்தோம்னா when glucose is not there. Okay, glucose இல்ல lactose இல்லனா, so when there is no glucose or lactose, is there a work for the yeast? No. So therefore it will not express. So no expression. Adhe, if you have only glucose, then also it is not going to express. So no work, it is not going to express. But when there is a lactose, okay, lactose irundaduna, of course lactose is the one which your yeast is needed. So lactose irundha enna pannu, it will express. So in the time la mattumda there will be transcription. So transcription nadandha RNA synthesis agum. RNA synthesis aanada protein code panna mudiyum. Protein da your beta galactomidase, permease, acetylase அப்படிங்கிற அந்த enzyme. So in the enzymes la எப்ப only when lactose is available your yeast is going to produce it. Otherwise it is not going to produce. So that is your gene expression. That is how your gene expression is done. Other than we are going to represent it by lac-operon model. We are going to express it by lac-operon model. Now what you do is, along with me, try to draw that lac-operon diagram. So what you're going to do, you're going to draw that lac-operon diagram along with me. So remember, you're going to have a big gene. Okay, just draw a big gene. In the gene la வந்து பாத்தீங்கன்னா first what you have is you're going to have a lac. Okay, you're going to have a lac. Okay, then what you have is you're going to have CAP site. What you're going to have a CAP site. Then you're going to have a promoter. Then you're going to have operator. So other lac-operon concept. So you're going to have operator. Then what you have is you're going to have three things: Z, Y, then you're going to have A. So in the Z, Y, A, these are your lac genes. So in the Z, Y, and A, so these are going to be called as your lac genes. Z and Y and A, this is called as your lac gene or lactose gene. Okay, this is going to be your lactose gene. So, but in the lactose gene, what it does is it produces enzymes that are necessary for your lactose metabolism. But adukku munnadi, you're going to have operator site, you have a promoter site, you have your cap site, you have your lac site. Idella enna pannudhu? it has some important steps. So LAC-I, it is going to stand for inhibitory. I stands for inhibitory. So what happens is these are nothing but your housekeeping genes. Na sonnella housekeeping genes. Ivanga da the LAC-I da, it is going to be a housekeeping gene here. So இவங்க என்ன பண்ணுவாங்கன்னா எப்பயுமே ஆக்டிவா இருப்பாங்க. So they are going to be active always, expresses 24/7 and this forms a repressor tetramer. So இது எல்லா டைமுமே இருக்கும். இந்த repressor tetramer என்ன பண்ணுனா, they are going to be binding with. So what it does, it is going to bind with your operator site. So this LAC1, it is going to bind with your operator site. Okay, so you are going to bind with the operator site. Okay, this is going to be, so இந்த இது வந்து பாத்தீங்கன்னா it is going to be having a repressor. This repressor, it is going to bind with your operator site. It is going to bind with your operator site. Then you have a promoter. So promoter enna pannu, as usual, what your promoter does, it is going to promote. How it promotes, inga da ungaloda RNA polymerase vandu okkarum. So RNA polymerase inga da vandu okkarum. Once it sits on this RNA polymerase, it is going to do a transcription, as I told you earlier. In the transcription நடக்கிறதுக்கு இந்த promoter phase-ல வந்து உட்காரும். அதே மாதிரி you have this repressor. Repressor is going to retard your operator. So ரெண்டு வேலை, promoter you are going to have, that is going to promote your operations. You are going to have repressor, that is going to repress your operation. அப்ப இந்த CAP site na என்ன? CAP site is nothing but this is the place where cyclic AMP complex. Okay, this is going to the place where you are going to have this complex. Is working clear? If you have any doubt till now, you can ask me. Next, what we are going to see is how this mechanism of action is done. Okay, I'll tell you what this mechanism of action done. If there is going to be, okay, if there is going to be a reduced glucose, if there is going to be less glucose, when there is going to be a lactose presence in the environment. So example for you, for the muscle, you are going to do a severe exercise. That particular point of time, what happens? You will not have glucose, you have lactose. Similar way, in a E. coli, in your lactone operand, what happens is there will be reduced glucose, there is going to be increased lactose. When there is more amount of lactose available, what it does is, okay, what it does is this lactose goes and directly bind to your operon site and it will be asking it to start the transcription. Whereas what your glucose does is, your glucose decreases. This will be increasing your cyclic AMP level. So once the cyclic AMP level is going to be increased, it forms a cyclic AMP and cap complex. This is going to, okay, come and attach this cap site. In the cap site attach panona, that is going to start your promoter RNA polymerase start panno. But unfortunately, you have one important thing called as opera. Opera is an operating site. Appa inge a positive stimulus. So promoter site a positive stimulus enna panona, in the opera remove panedo. Opera or block between your promoter site and your LAC genes. And the rendu perku nadula irukra or component ana opera nama remove pandrom. So now what happens? Your opera is removed. So opera in the particle remove aidide. Appa in the nadula opera remove aidchna enna agum? Your promoter site is going to go to your ZYA, and this ZYA is going to produce the enzymes of lactose. This is going to produce the enzymes of lactose. That means your gene is going to be turned on that particular site. In the particular site long a gene turn on pandrenga. So if a gene turn on, switch on, so your transcription will be starting here. This is how your transcription is done. I hope you can understand. Lactose glucose. What happens? Lactose is going to, okay, now you are going to talk about lactose as well as glucose. Now what you have both lactose as well as glucose. Now what you are going to do is, now you have both lactose and glucose. Lactose increased. So what happens? There is going to be a it is going to go and sit at this opera and try to push that opera outside. At the same time, glucose is increased, therefore cyclic AMP level is decreased. Cyclic AMP level decreases, you won't have the cyclic AMP cap complex. When there is no cyclic AMP cap complex, it will not help in removal of your opera. Therefore, the transcription does not occur. Even though you have more lactose, if there is more glucose also, what happens? you cannot remove that opera site, then what happens? Transcription does not occur. Transcription does not occur. This is how your lac operon model is going to work. If you have any doubt in this lac operon till now, you can just put in the chat box. Now I will tell the inhibitors of transcription. I told you 4 things. Again, I will repeat it for the sake of everyone who are there in the who are watching this session. So actinomycin D, it is going to bind with your DNA strand and it is going to block your transcription. Rifampicin is nothing but your TB drug. This is going to block your RNA polymerase. Alpha amantine, again it is going to block your polymerase. Alpha amantine is nothing but your mushroom poisoning. And you have this 5-deoxyadenosine. This will inhibit the splicing mechanism, and this is a synthetic drug. Okay, this comes to the end of the inhibitors of transcription. This comes to the end of inhibitors of transcription. Now, at the beginning, I told you some non-coding drug. If you remember, some non-coding RNA, sorry, not a drug, or non-coding RNA. What are the 2 RNAs I told you? Number 1, microRNA. Number 1, I told you about microRNA. And number 2, I told you about small siRNA. Okay, this microRNA, what it does is, it is going to, it is going to modulate all this gene expression. If you gene expression பாத்தீங்கல்ல, இந்த gene expression humans-ல யார் modulate பண்றாங்க, ஒரு human being-ல யார் modulate பண்றாங்கன்னு பார்த்தோம்னா உங்களோட microRNA. The microRNA is the one that modulates your gene expression. Okay, va? So how does this modulate? So you will be having a pre-microRNA. You are called it as pre-microRNA. This pre-microRNA in the presence of dicer nuclease. So in the presence of dicer nuclease, what it does is it forms pre-microRNA. Pre-microRNA gets converted into pre-microRNA. So in the pre-microRNA, again in the presence of drosha. So these are some enzymes. Okay, these are the enzymes named after some scientists, named after some Greek or Latin words. Namakku theva and the word mattumda. So dicer or nuclease converts your pre-microRNA to pre-microRNA to pre-microRNA. Pre-microRNA gets converted into microRNA in the presence of drosha. So in the question, unfortunately, no one expected. This was asked in the INICET. Okay, your AIMS exam. So AIMS entrance exam la INICET la they have asked this particular enzyme name. Similarly, microRNA is going to be modulating your gene expression. This is also one question asked in your INICET. So those who wanted to go to pursue your PG in your central institute, you wanted to remember this. Similarly, you have this siRNA, isn't it? In the siRNA enna pannuna, they bind to mRNA, and finally what it does, it is degrading it. So the one which kills your mRNA is your siRNA. Okay, they are going to degrade your mRNA. Okay, that's degrade on a product, we call it as P bodies. The degrade on a product of what we call it as P body. So this non-coding RNA is otherwise called as your interference RNA. Interference RNA, otherwise called as iRNA. So iRNA enna panna inclusions form panno. It forms inclusions. That is okay, inclusions form panno. That is going to be your iRNA or interference RNA. Are you clear up to this? Now, can you just, I'll just give some questions to you. I'll just ask some questions to you. Those who are in the YouTube, you can just put it in the chat box. Number 1, linkage protein. The linkage that is going to be between 2 nucleotides. What is the bond that is going to be present between 2 nucleotides? Someone, can you put in the chat box? The one that is going to form a linkage between 2 nucleotides. That is called as, I'll write it down. Linkage between 2 nucleotides. What is the one that is called as linkage between 2 nucleotides is called as? Linkage between 2 nucleotides is called as, this is like a revision for you. I'll give the answer. I'll go to the second question. Okay, I'll give you some statement. Which are the following are true regarding eukaryotic transcription? RNA polymerase does not require template. Is it true? RNA polymerase does not require template. Is it true? Second, all type of RNA are synthesized in the nucleolus. all type of RNA are synthesized in nucleolus. No, only one type, RNA polymerase 1 is alone synthesized in nucleolus, rest all synthesis in their nucleus. So that is wrong. RNA synthesis requires primer. Do you require a primer? No, RNA synthesis does not require a primer. And last option is phosphodiester bond formation occurs due to the pyrophosphate hydrolysis. Yes, so pyrophosphate hydrolysis. So all these questions I'll again put it in the chat in your WhatsApp group also. Kindly answer those questions. Now what I will do is remaining questions I'll put in the WhatsApp group. So what we will do is this codons particular topic alone I have left it for the next day, tomorrow. So tomorrow what we will do, we will go for the next part. Okay, so I will put all the questions. So what I will do is now I'll just take your phone. Already I have given some questions for you in the group, isn't it? I'll tell the answers for that. What is true about histone acetylation? Okay, what is true about histone acetylation? Almost 20 gave answer as increased euchromatin formation. Okay, 20 gave answer as increased euchromatin formation. Second option was increased heterochromatin formation and methylation of DNA replication. Remember, histone acetylation increases euchromatin. So option 1, increased euchromatin formation is right. I have just answered the answer also. I'm just putting down. You can see my answers in the group so that it will be helpful for understanding. Methylation of bases in DNA usually. I've given 4 options. The answer you have given, what is the answer you have given? Facilitates the binding of transcription factors to DNA, makes the difference in activating only of it occurs in enhancer region, inactivates DNA for transcription, results in increased production of product in whatever gene is methylated. So methylation of bases, I told you, methylation will increase the heterochromatin. If it increases the heterochromatin, what happens? It inactivates the DNA transcription. Mitochondrial DNA is a circular DNA. That is easy. I don't know, only one I've answered it as a ZDNA. That is wrong part. Helical structure of DNA was discovered by Watson and Crick. And DNA replication occurs at which phase? S phase. That is 45 have given the right answer. Everyone have given right answer. Very good. Gap filling is done by, now I told you again, I have shown you a video. Now can someone put in the chat box? Gap filling is done by DNA polymerase 1. And linkage between 2 nucleotides, I told you, it is phosphodiester bond. And all are true regarding mitochondrial DNA except it contains 37 genes, transmits from mother to offspring. It causes Leber hereditary optic neuropathy. Exactly. So almost everyone gave a right answer for it. Very good. So today we have completed, we have revised replication. Then we have revised DNA repair mechanism. Then we moved on towards the types of RNA. And then we have talked about the transcription. So tomorrow we will have 2 sessions. In the morning, what we will do is, we will complete the translation. We will complete what is codons and we will talk about the translation, okay, about the mutations tomorrow morning. After completing the mutation, after completing the translation, in the evening, almost in the night, what we will do is, we will almost in the evening at the same time, we will talk about the molecular biology techniques. With that, we will be completing entire molecular biology. So more 2 topics we are left with and some minor topics, which we will be completing tomorrow. After tomorrow, you can close your molecular biology. And from Monday, what we will do is, we will start with this glucose metabolism, carbohydrate metabolism. We will take only 2 days for carbohydrate metabolism, Monday, Tuesday. 2 naalla carbohydrate metabolism complete pannidala. So after that, you have this amino acid metabolism, fatty acid metabolism. Along with that, next week, we will complete entire biochemistry. So next week, our target and entire biochemistry must be completed. Okay. So thank you so much for joining today. So we will be meeting you tomorrow morning again. Okay. Thank you.